Hello everyone, you are all welcome to lecture number 1004, and today's topic is the biochemical methods of purification of industrial effluents, or we can call industrial waste. When we say biochemical, it means reactions are involved, microorganisms, bacteria are involved, reactions are there like oxidation. So all these processes are involved for the purification of industrial effluents, and there are mainly two methods of this biochemical purification of effluents. One is aerobic, that is in presence of air or oxygen, and the second one is anaerobic, means that is in absence of air. The most popular and widely used method is aerobic, means in presence of oxygen or air, the oxidation of all the impurities, whether it is inorganic, organic, salts, mainly sulfides, ammonia, they are all broken down by bacteria and selected microorganisms into non-toxic or into some very simple chemical substances. And this can be determined by measuring the factor that is known as BOD, that is biological oxygen demand, COD, that is chemical oxygen demand, MTC, that is more temperature control. So these factors are the parameters to determine the purification of the water, and the biological demand and chemical demand of oxygen plays very important role. And if it is not fully purified, there will be lot of biological and chemical demand, and that ratio should be maintained. If it is not done, all these factors, then the aquatic life will be in danger, and even that water is not safe for human consumption. So these factors I am going to discuss one by one. So in the case of biological purification of waste or effluents, as I have already told you that in this microorganisms they play a very important role as a bacteria, and these impurities, they act as their nutrient, source of nutrient, and in this way the biological oxidation will take place, and the most important ratio in that biological oxidation is that is BOD, biological oxygen demand, COD, that is chemical oxygen demand, and that will indicate. So here number one, biochemical purification of industrial effluents. This is a large scale and the most widespread method of purifying industrial effluents and domestic facial sewage. This method is mainly used for waste which contain organic matter, either as an independent method or in combination with other methods. The method is based on biological oxidation of organic and some inorganic substances due to the action of microorganisms, using the impurities as a nutritive substrate and forming harmless oxidation products such as water, that is H2O, CO2, nitrate and sulfate ions. So once the microorganisms, bacteria, once they will act, and with the help of the activities of these microorganisms, those effluents, means industrial waste, will be broken down into harmless substances like water, carbon dioxide, nitrate, and sulfate ions. The biological purification consists in destroying organic and some inorganic products, pollutants, using a microorganism culture of specific nature. So this is very important for destroying particular or specific. We are using the different type of very specific culture to destroy the very specific type of effluents or industrial waste in nature by using bacteria. It is possible to oxidize most of the organic impurities. The degree of decomposition of organic compounds in biological purification is characterized by the ratio of BOD, that is biological oxygen demand, and COD, that is chemical oxygen demand. This is very important point to note, that the greater the biological oxidation, if there is in huge quantity the biological oxidation, that is called, the greater the biological oxidation of a given impurity, the higher is the ratio of BOD and COD. Substances for which this ratio is greater than 0.6, it means such as aldehydes, alcohol, acids, anilines, and ethyl acetates are decomposed, and then the ratio will be 0.6 BOD. So in the process of purification of the effluents by a biological process, two factors are very important, that is MPL and MCT. And here I have discussed this. Number 2 is the MPL. MPL means maximum permissible limit. So there is a limit given, and that is the maximum. After that we cannot allow, or WHO cannot allow more than that. That is called MPL limit, that is maximum permissible limit of matter to be subjected to biochemical purification in gram per m³. First, for organic acids such as cyclic or butyric acid is 100 to 500. The alcohols and ethers are 300 to 500. Number 3, ions of metals such as zinc, nickel, and chromium is from 1 to 10. Number 4, phenol is up to 1000 limit. Biological purification requires strict observation. Especially strict observation means that is temperature, pH is very important, and if it is not properly maintained, then the biological purification is not possible. So the observation of the technological required regime, because the vital activities and high efficiency of microorganisms are possible only under definite conditions, namely a temperature, and temperature should be 30 to 40 degrees centigrade. And if it exceeds more than that, then the activity of microorganisms will diminish, and if it goes above 50 degree, then they are totally ineffective, or they may be destroyed or killed. So the best temperature to do the biological purification is from 30 to 40. It should not exceed more than 40, and the pH should be 5.5 to 5.8. That is, we can say it is slightly acidic, and with normalized concentration of the substances having harmful effect on the microorganisms. Number 3, the MTC, that is maximum tolerated concentration, that is called MTC. That is very important also, that how much temperature can be tolerated by all these biological impurities and their purification. So the maximum tolerated concentration of individual substances is a mixture of fissile sewage and effluents prior to biological purification in milligram per m³ are as follows. Phenol is 1000. Second, formaldehyde is 25. Third, synthetic detergents, or we can say biodegradable detergents are 20 to 50 value MTC. Number 4, methanol is up to 5000 MTC. Cyanide is only 1.5, and the mercury is 0.005, and mineral salts not greater than 10 gram per m³. When the concentration of the impurity is greater than MTC, for biological purification, the effluents are necessary to be diluted before being fed into the purification unit. So once you are going for biological purification, that is biochemical purification, where you are going to use some bacteria or living organism, that is microorganism, if these parameters, all these values, if it is not like that, you have to check, and if it is more or concentration is more, then you have to dilute it. Otherwise, the biological purification will not take place. So this biochemical purification of industrial effluents or waste is very, very important, and globally it has a lot of impact, and mostly people are using this bacteriological method of purifying the industrial effluents. With this I will stop. Thank you very much for watching the lecture.